Hey guys, Fatato here, and I'm going to talk to you a little bit about the structure of DNA today. You may be asking what this is next to me, and I'm going to start by telling you a little bit of a CV history story. I coached the varsity girls soccer team. I was the varsity girls soccer assistant coach for 10 years, 11 years. And so one year what I did was when we were on our way to the playoffs, once we started playoffs, I started to build this ladder. And the conversations that we had as a team was about how in order to get to the top of a ladder, you have to take one step at a time. So each of the rungs of the ladder represents one of the games in a playoff game. And you can't see the whole thing because I'll hit my ceiling, but like it starts way down here, okay? And each rung has the game, who we played. It has all the signatures of the players and then the score. And this was the year that we made it to the state final, and we won the state final, 1-0 CD versus Penn Manor. So I didn't have a real ladder in my house, like a big enough one, but I had this. So this is what I'm going to use today to talk to you about DNA. Hi guys. All right, so let's talk about DNA. DNA, as you've learned, is shaped like a double helix, which is a twisted ladder. It's called a double helix because it has two sides, one side here, one side here. They're held together in the middle. All right. And we'll talk about that. And the video you watched talked a little bit about that already. And then it's twisted. Obviously, this isn't a twisted ladder. So it is a double helix, but it's not twisted. It's double stranded, but it's not a twisted ladder. Just because that would be harder for us to see stuff. So the shape is a double helix, which means a twisted ladder. Okay. Now, at the end of your last video, you learned that DNA is a polymer. Polymer means it's made up of many parts. Poly means many. And polymers are made up of smaller pieces called monomers. Mono means one, so it's single or it's small. Monomers for DNA are called nucleotides, okay? And nucleotides are made up of three things, okay? The first thing that a nucleotide is made up of is a sugar. So right here on this ladder, I have drawn an S and it's red, and that represents sugar. And the specific sugar that is in DNA is called deoxyribose. That's actually where the D in DNA comes from. So sugar is one part of a nucleotide, deoxyribose sugar. The second part of a nucleotide is a phosphate or a phosphate group. And so I have that represented up here with a green P. So two of the three parts are deoxyribose sugar and a phosphate. The third part of a nucleotide is this right here. And this B represents base. We'll talk about the 4 different bases in a moment, but the base is the third part of a nucleotide. These are often referred to as nitrogen bases or nitrogenous bases because they have nitrogen in them, okay? So the 3 parts of a nucleotide are deoxyribose sugar, a phosphate, and a nitrogen base. Okay. And these are the monomers that make up DNA. These are nucleotides, sugar, phosphate, base. All right. You'll notice that the sugar is always attached to the base, and that is correct. The sugar is always attached to the base. Now, nitrogen bases. The nitrogen bases are adenine, thymine, cytosine, and guanine. And according to Chargaff's rule, which you have already studied, remember Chargaff, in case you're wondering, he's my favorite scientist. I want to one day have a dog named Chargaff. Okay, I just really like that name. So A pairs with T and C pairs with G. So if this was an A, this base over here would have to be a T. If this base was a C, this base over here would have to be a G, and vice versa, because they can go in either direction. So the nitrogen bases are A, T, C, and G, adenine, thymine, cytosine, and guanine. So let's talk about the rest of the whole overall structure of a ladder. A ladder has sides, which are sometimes referred to as the backbone. And the sides of the DNA ladder are alternating sugar, phosphate, sugar, phosphate. Let's do that again. They're alternating phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. And the other side would actually be opposite. It would start sugar, phosphate, sugar, phosphate, sugar, phosphate. we'll get into the details about that in class when we're together on Thursday. So the backbone of the ladder, the sides of the ladder are alternating sugars and phosphates. That means that the rungs, and rungs are where you put your hands and feet, the rungs of the ladder are our nitrogen bases. Okay, now I'm going to grab a marker real quick. So the rungs of the ladder are the nitrogen bases, but something has to hold the nitrogen bases together. So what would be right here would be bonds. Sometimes there would be 3, sometimes there would be 2, okay? And the other base would be like this, because they match up, their shapes match. So let's go back and let's say this is an A, this would be a T, okay? You see that? These bonds that hold the nitrogen bases together in the middle, where the rungs of the ladder are, those are hydrogen bonds. Hydrogen bonds, okay? Sometimes there's 3, sometimes there's 2. So the backbone or the sides of the ladder, alternating sugar phosphates. The rungs of the ladder are your nitrogen bases are your nitrogen base pairs, and they are held together by hydrogen bonds. So make sure you answer the questions in your hyperdoc. And on Thursday, we're going to be doing some more in-class or together as a class drawings and diagrams and things like that. So don't forget to do the very final portion of your hyperdoc, which is a picture. Remember, it doesn't have to be professional. I can't draw. Most of you guys know that. So we're just looking for your greatest and best effort that you can give us. All right. So hope you guys are doing well. Hope this all made sense. If you have questions, send them to us in Schoology, or you can ask us in class. See you guys.